Okay.
Sounds good, Bruno. I'm going to go off, stop video until I get started. Thanks.
Yeah, and then during your presentation, clearly keep, if possible, keep also the video on so people can see you. And then after, when you are not speaking, better maybe if you keep clearly the microphone off and also the video off. And usually,
Bruno, sorry, I have to switch off the camera while I'm speaking because my connection is not so good. I prefer to have a good voice.
You can do like that, okay.
Okay,
and,
make sure your screen is sharing though.
Yeah. And then at the end, before everybody leaves the webinar, please remember that we have to take a virtual picture, group picture, and some things that we have done, and it's always nice. So I will ask to turn on camera, and we will take a virtual picture if you agree to do that.
Sounds nice.
We are finally getting closer to normality.
I recognize that voice from last night.
Stayed at home a lot and kept to social distancing. No traveling.
Is that Joe telling he's joined us?
No, sorry, that's just me loading the film just in case there's a problem. Has Joe managed to work the film?
Maybe, I don't know. Have you sent the link to...
Is Joe with us?
I just received an email.
Do we have Joe or not?
Well, I think that we should start, however, because we have already a good number of people connected.
So should I start by sharing the screen, or are you saying something first, Bruno?
Yeah, no, well, a few words just to say welcome to everybody. I want to thank Hilde, you and Jeremy for organizing this webinar. This is webinar number 7 of a list of webinars that we are organizing with the iPlanta network. iPlanta is a cost-supported project. The cost is a EU program on cooperation science technology, and the iPlanta is a specific network related to RNA technology. And today webinar is specifically related to communication, and I'm really happy, proud to have such a good number of people and very well-known people like Mark Linus, and welcome Mark. But besides that, also Patrick and all the other people that are invited today for this webinar. So you already know more or less how it works. Hilde, Guy, you and Jeremy will manage the different section. I asked to stay more to keep the timing, so not to have too long webinars, but at the same time, if you have a nice discussion and people want to interact, don't worry, keep your time. As already said for everybody, I'm recording the webinars, and the file will be posted on the iPlanta webpage. At the end, we will take a joint photo for all the people that virtual photo for the people that will be still connected. So thank you again, and now I give the floor to Hilde for starting today's webinar. Okay, Hilde.
Thank you, Bruno. Can this is up there now?
Yes, perfect.
Very good. Joe just sent me a text saying that he can't find the invitation. So if you could please send it again, Bruno, so he gets to join us. Thank you. Okay, Well, a heartwarm welcome to all of you. I'm really proud and really happy, and thanks to Bruno to encouraging it and having a good process. I think there is some upsides to this new COVID-19 world, and one is that we can get the best speakers because it's we have no borders. It's a short distance. We can have a larger audience, which is also good, and it's efficient, and we can all work together. But I hope we will work together to open the new world, as Joe will say, show in his presentation. Okay, I was thinking first, okay, so just for those not knowing RNAi, just shortly say that RNA interference was or is still a difficult term, and before the COVID mRNA vaccine talk, most people didn't know what RNA was either, but that has been easier now when most journalists knows what RNA is somehow. And this really easy sketch is just first to say that we are all a sum of genes or cells, and those cells are differentiated, and they are as they are due to the genes active in the respective cells. And RNA is a link between DNA and protein. So we can see RNAi as a regulator, and we can say it can be turning genes on or off, but more correctly fine-tuning, so the genes can be more active or less active. And RNAi was first discovered in plants, so of course for our community that is especially exciting, and it's used in food production and in medicine. And now with vaccine interest for COVID-19, RNAi could be a better solution than mRNA, since it's more stable than mRNA. And we will get some of that in some of the presentations. And we can also see RNA as the cell's initial protection against invasions from viruses, for instance. So that makes a natural link between vaccine production and the use of the biological system or techniques when we take advantage of it in the lab. And this is just linked to a nice short cartoon or interviews with good scientists telling about the RNA history. I will mention it shortly also in the communication session. So the difference between health and food production is that food is culture. And one of the questions we have today is whether the COVID-19 can change or open the minds of regular users for using the technology also in food production. And in communication, relevance is paramount. And now with this new interest of or belief or recognition that we need science and also RNA, maybe that could open the future for using the method in food production. That's one of the things I hope for, but maybe I'm too patient because we've been hoping for many decades. So one of my easy or simple questions is whether the impossible could be possible also for Europe. And we will get back to the Impossible Burger story in the communication part. And this webinar has been set up so there are 3 sessions with 2 main speakers of 15 minutes, then short introductions of 5 minutes to stimulate a discussion. So I sincerely hope that many people will put questions in the chat, and that we will get vivid discussions between the sessions and at the end. So stay put and use the chat. Thank you. Then I guess I'm going back to Belgium so that he will put us somewhere else.
Yeah, thank you, Hilde. Is the sound clear for everybody?
Very good now.
Okay, good afternoon, everybody. Like already introduced by our coordinator Bruno and the organizer of today, Hilde, we have 3 sessions of today focusing on communication in relation to RNAi technology. And the first session is where we selected, in fact, 3 groups to have an idea about their perception on this RNAi technology. We have 3 speakers, and the first speaker is Mark Linus, and he will give his a view on the public perception. Now, as an intro, short intro, Mark is from Britain. He's an author, a journalist, and an activist. Maybe some of you know his famous book, The God Species, Saving the Planet in the Age of Human, of 2011. And of course, he's also well known for his opinion on GMO. And that's also specifically the reason why we invited him to give his introduction here, or to give his opinion here about the users group perspective and the public perception specifically on GMO and RNAi technology. Mark, you are present?
I'm here, absolutely.
Yes, so
Ready when you want me.
I give you the floor, and you can bring your mission to the audience of our group here. We are very close to 100 people now, so I think you can broadcast what we got wrong about GMO crops. Please, Mark. Thank you.
Okay, there we go. Can you see it on the screen now? Just checking that works. And you can hear me fine. Even better. Okay, so I'm going to share with you some of the lessons that I think I've learned from the GMO experience. And so that's by way of analogy, really, in that with RNAi, you're talking about a new technology, and obviously one which nobody has an opinion of yet. So what can we learn from the way that it all went wrong with GMOs? Now, I just want to share with you some real world experiences of why and the GM debate went so badly wrong and why it matters. This is a picture that came from Tanzania of one of the trial sites being burned, where they were testing GM drought-tolerant maize. This actually, you may have heard more recently that Tanzania has banned all of its GM trials altogether. So basically, the at least transgenic aspects of biotechnology have basically ceased to function because of the I would characterize it as anti-science opinion of the government. But this was even earlier than that. So after trials finished, all of the transgenic material has to be burned because it's seen as being somehow posing a significant environmental risk. Now, this is in a country where you've got serious drought problems already, almost certainly related to the accelerating impacts of climate heating. And from what I could see, and there's a picture of me and one of the researchers, Nicholas Nyange, on site in Makutupora, which is the GM test site in the central part of Tanzania. These were the best and the healthiest looking maize plants that I saw throughout my entire trip to Tanzania. Most of the rest of them are stunted. They're very low yielding. And here's, in fact, this has the BT insect resistance trait as well as the drought tolerance trait. And you know, these were fantastic plants, but they have not been, to this day, there's no farmers anywhere which are allowed to grow them because of the GM thing. And you know, it was all over the place, the kind of the specific attitudes to GM. So all the regulatory stuff, this is a sign that was outside the test site, you know, not for human animal consumption, no unauthorized entry. All this kind of stuff adds to the kind of optic that something strange and potentially risky and dangerous is happening behind those walls. And those kinds of things are really, really important. And I want to share with you just a quick précis of my own life story. I'm one of those people in white there back in 1995. At that point, we were destroying an oilseed rape crop that was part of the government's farm scale trials. And this, yeah, we successfully did that. And not only did we destroy this crop in this field, but we also destroyed all of the transgenic experiments throughout the entire country. And that's basically put a stop to GM crop research in the UK up until comparatively recently. We're against pretty much all aspects of biotechnology. We tried to steal Dolly the sheep even. This came out, I wrote about this in my Seed to Science book, and it came out a bit in the papers as well. But I think I just used that to illustrate the fact that we were against cloning, we're against, so it's not the specific, nothing specific about the kind of conventional GMO transgenics that was what excited our opposition. It was against the whole broad spectrum of the way that the technology, or the genetic technology as a whole has been rolled out. And I think that's important to understand because it will include RNAi as well, of course. I won't go into too much detail about this, but the opposition to genetics as almost in totality goes a long way back. The first anti-GM protester or organizer I could find is a guy called Jeremy Rifkin, who's still active, in fact, campaigning in favor of hydrogen and renewables and a lot of conventional and environmentalist type stuff. But he was onto this way back in 1977. So more or less in symbiosis with the invention of genetic engineering as a technology, the forces of opposition were also mounting. And Jeremy Rifkin then convinced his friend Benny Howlin from Greenpeace in Europe from 1995 onwards to campaign against genetic engineering, and the rest is pretty much history. Greenpeace made the campaign global. I got involved actually after attending a Greenpeace sort of organizing session in about 1996, I think. And that was a huge, huge success. The results, which obviously are still with us and to the present day, are that GM is effectively banned or at least regulated out of existence in all of Europe, most of Africa, and most of Asia as well. So pretty much the only places where GM crops are grown on a substantial scale are North and South America, and for largely commodity crops too. The reason why this happened, I think, and having looked at it, is that the way that GM crops were originally rolled out. So this was on the heels of Silent Spring, by a company which was primarily involved with selling pesticides, of course. Monsanto's interest in the first trait that was deployed for GM crops, the Roundup Ready soybeans, was of course to sell more of its patented Roundup and to sell patented seeds alongside in a package. And to us as environmentalists, this was everything that was bad about modern science, was that it would enable big multinational corporations to worsen the worst aspects of monocultural industrialized agriculture, which were so well exemplified by the trait of Roundup Ready. And this led to a perception which I think was very widely held, that Monsanto was the world's most evil company. And there's no way back really for GMOs as a whole, even though a couple of years after Roundup Ready was deployed, BT was rolled out too. And had that, and I like to conduct a kind of thought experiment. If they'd started with BT, and that was had a primary aim of reducing pesticide use or insecticide use specifically, maybe we would have been in a different place. But because the whole debate, the narrative of the debate was framed around the fact that this dangerous thing was being done to the genetics of our food in order to put more chemicals on it, there was simply no way that any scientist could make an effective argument in favor. And even though I had a change of heart myself, looking at the scientific consensus on the safety and environmental friendliness overall of genetic engineering, it's not like, I don't feel like this has been something which has, part of a broader change, if you like. I think it's still there's still a very much an anti-GMO narrative, which is predominant in society. And I got a lot of flack, of course, about this. I was immediately called a sort of Monsanto shill, all that kind of stuff. And while there are examples, and this is one I've been to Bangladesh, I've visited with many of these farmers. They're using BT traits in eggplant, so in aubergine, to resist pests and therefore to dramatically reduce pesticide use. It's now too late though, and the environmental and anti-GMO groups will not accept this as a technology, even if it demonstrably helps smaller farmers and reduces chemical use, because their opinions have been formed, and they were formed 15 years prior to this event. And the public as a whole also remains extremely skeptical. There's not a single country where more people believe that GM crops are safe than unsafe. The United Kingdom is sort of somewhere around the mid-range. Australia, people are roughly evenly split, but in some countries they're very, very anti indeed. And that includes some of the most influential big countries like Germany, Italy. So most of the major players in Europe are very, very anti to this day, and likely to remain so. You mentioned, Hilda, at the beginning about whether COVID vaccines maybe would reframe the debate, at least about the use of RNA, because of course biotechnology in different forms has been used all the way through the very rapid and successful effort to deploy vaccines against the COVID pandemic. I think I'm a little bit skeptical about that, and I'll talk a bit more about that just in a minute. But I think we need to learn from the lessons of the GMO debacle and try and figure out how we can look at ways in which RNAi as a technology might be employed in a better and perhaps more favorable way. So there's two things I thought it's important to remember about RNAi when engaging the public in debate. Number one, nobody knows what the hell RNAi is. And number two, no one knows what the hell RNAi is. And that's not going to change. Nobody knows what RNA is, even though you said they can use the word or the phrase, people can say RNA vaccines, but nobody, I mean, I've described to numerous journalists over and over again how RNA works in cells, a sort of central dogma, if you like, and they quickly forget, because people don't have basics of, any kind of basics of cell biology in their heads. So even though you might learn this in high school, you forget as soon as you've left high school, and that's basically where the situation remains. So what can we do about this? What are the lessons we can learn from GMOs? Number one, the antis will hate it before they even know what it is. I think this is very clear from the experience we've had over genome editing in Europe, which has already been characterized as GMO 2.0 by all of the anti-GMO groups, and that includes the very broad agroecology stroke organic movement. And they were successful, obviously, in getting a European Court of Justice judgment, which stuck gene editing in this pretty much the same regulatory basket, prohibitionary regulatory basket as GM crops back in, I think, 2018. And that's where things have become stuck. And it's difficult really to see a path forward for gene editing in Europe, really, because it's already been caught up in the GMO mess. Now, when you're introducing a new technology, you have an option, obviously, to try to frame a more positive narrative, and optics matter on this, and in particular, who's developing the technology and in whose interest is it being deployed, and it really matters how this is seen. And let's look quickly at what I mean about setting a narrative. Here's an example of a good narrative. This is an environmentally friendly new technology, environmentally friendly new farming approach. I wouldn't use the term technology, actually, which uses the power of nature. So it's natural, and it uses biology to control pests and produce better crops without using chemicals, without using chemical pesticides in particular. Now, a bad narrative, we could take this from pretty much how we started with GMOs. This is the scary new technology that's like GMOs, which we already know are bad, but it's even worse than GMOs because it's more complicated and therefore potentially has lots of horrible impacts that we can't anticipate. And we won't know about these yet, but we'll only find out when it's already too late. So we'd be better off, and this is how things are always characterized, we'd just be better off being organic. Why can't we go organic? We can stop using the chemicals, we can still have plenty of food. Why wouldn't we do this? And that's always the kind of counter narrative which is going to be presented. Now, just want to put together a few kind of half-formed thoughts here about how we might move forward here. I think it's probably important to understand that if we even start with a situation where RNAi is characterized in the same basket as GMOs, then you're doomed from the outset, in particular in Europe and Africa, but probably a good deal else besides. It's important, I think, for the scientists among you to understand that no amount of scientific studies of all of these things, like off-target effects and how it might affect the diet, diets of vertebrates and whatever, will satisfy the opposition. Will satisfy the opponents. So it's really important to understand that the opposition to GMOs and opposition that will manifest to this is moral and absolute. So technical arguments about safety, when they're put forward by opponents, is simply a way to manifest their total opposition without having to challenge the dominant scientific paradigm. So what I mean by that is if they argue that it's unsafe and they come up with some pseudo-scientific studies, then they can continue to block it without having to ever admit they're against it for essentially religious, moral, religious reasons. And I think from that, you can learn that if opposition engages moral intuition, so must proposition. So the beneficiaries must be clear and deserving, things like the environment or poorer farmers, not shadowy figures in lab coats and greedy multinational companies. You can see that referring pretty clearly to, again, to the GMO experience. And I don't think it's going to be possible to get away from fears about RNAs changing our DNA. This is what's already been the case with COVID vaccine misinformation. And I've had countless emails from people, because I've written about COVID vaccines, saying, won't this change our DNA? Except this time it's going to be published on the front page of The Guardian rather than being banned by Facebook, because The Guardian has an ideological bias in favor of organic farming and against biotechnology. So you're going to be hit by that for sure. And just to finish up, how might you try and move forward here? I mean, it's certainly worth considering having some kind of industry-wide discussion without kind of forming a cartel, obviously, but at least if you try to ensure that the first mass market application has clear benefits that promote and frame a positive narrative. So, I mean, obviously, the primary way that RNAi, as far as I can tell, is proposed to be used, at least in food and agriculture, is to reduce pesticide use. So a different way of biopesticides, if you like, particularly if that's not transgenic, so isn't delivered inside plant tissues, but is applied from outside, then that could be non-GMO, and it can be natural, and it can be biological even. But you'd need to be very clear about what the potential reduction is of chemicals, or even if you could say this is like organic in that it's absolute, you could do this without using any pesticides at all. And that's one way, obviously, to frame a positive narrative. And secondly, I think it's important to try to establish a scientific consensus, if one exists, genuine with, I mean, with the genuine sort of direction of the scientific literature about the safety issue in particular, and to ensure that institutional centres of expertise, like in the UK, it would be like the Royal Society. In the EU, of course, you've got all the science academies in all the different member states, and also at the EU level as well, who can serve as third-party validators and talk effectively about how this technology works, and in ways hopefully which are positive to the science. I think just let's not be too naive about how debates work and how it can be fairly negative experience being involved in public debates. Often these are very influenced by NGOs and involve things like citizens' juries being set up, which are always very biased because of who gives them evidence and who constitutes them. And they often start off with the narrative, a very anti-oppositional narrative. And at the end of all of this and this huge effort to engage the public, still nobody will understand what RNAi is. There you go. That's how I'm finishing. Thank you very much.
Thank you, Mark. Of course, everybody can bring in comments and questions in the chats, and then we can collect this for feeding the discussion afterwards. We selected a second speaker, Vera Ventura. She's a professor at the University of Milan. She's an expert in agricultural economics in the Department of Environmental Science and Policy in Milan. And she will bring a view on the European stakeholders. Vera, you have the floor.
Thank you, Guy. Yes, here I am. I just switch on the camera, off the camera, because I want to be sure that the connection is good, and then I share my screen. Can you see my presentation? Can you see it?
Yes, we can.
Yes.
Perfect. So,
Perfect.
thank you so much. Good afternoon again. I'm Vera Ventura. I'm the chair of the iPlanta Working Group 4, which deals with the analysis of the socioeconomic impact of RNAi solution, but also on stakeholders' involvement and networks. So my intervention will be focusing on the outputs of a very special event that we organized to meet stakeholders and policymakers. So the event took place in 2018 in Brussels, with a morning session at the Copa-Cogeca headquarters and an afternoon session at the European Parliament, with a very specific aim to create links with stakeholders, to inform them and introduce some results on RNAi solution, and above all, to open the discussion on the potential use of these new products. So the event had a large participation of stakeholders from industry, institution, growers and growers association, cooperatives, research institutes. And the meeting was opened by a brief presentation of what is RNAi technology, its application, and the specific activity of each working group of the iPlanta cost action. Well, many stakeholders expressed a great interest towards these innovative solutions, especially for topical RNAi application, which is, let's say, the most innovative approach. Even if a maize grower representative said that, in his opinion, farmers would prefer stable expression in plants with GM, because in his perspective, it is much easier to use with no need for treatments at field level. But more in general, many questions were about the practical use, the convenience and the efficiency of this new product. A stakeholder asked for the main shortcomings of RNAi technology, including the risk of resistance, but also what is the cost, how many treatments are needed, and so on. And one of the major concerns was the regulatory context of these new products, as well as consumers' acceptance, because farmers and producers are fully aware that without consumers' acceptance, there can be no market for these products. Then the afternoon session took place at the European Parliament, and again, after a brief informative introduction on RNAi-based new products for plant protection as alternatives to pesticides, actually some farmers' representatives had the chance to explain to policymakers the challenges related to the future of farming in Europe. They say that farmers need answers and need solutions to face the decrease in funding, the active ingredients ban, climate change and the need for more resilient plants, as well as the market, the volatility of the commodity market. So farmers asked to policymakers for open access to all the tools available in the innovation toolbox, including biotechnology, free research, open field testing, and so on. And then the final intervention was by a member of the European Parliament. He said that he's convinced that technology is the key for the future of agriculture and its productivity, but referred to the existing political concerns about climate change, he also noted that agriculture and food production are considered to be the bad guy. But he also said that this could be an opportunity to consider this new technology like RNAi as part of the solution, showing the benefits of RNAi-based approaches. So his main message was sell the benefit first, show the benefit first, pointing out and stressing the fact that good communication is crucial. And so I'm not a communicator, but I believe that this suggestion can be very relevant. And in conclusion, we can say that the 2018 event highlighted the need to continue the discussion. And for this reason, we are going to repeat the event with an enlarged networks of stakeholders. The webinar is scheduled for the 4th of March. And I'm pleased, it's a pleasure for me to, for all those who are interested, to invite you for participation. And thank you so much.
Thank you, Vera, for giving this information. Like I said, we selected 3 kinds of perception: the public perception and perspectives by Mark, the European stakeholders' opinion about RNAi solutions by Vera. And then we have a 3rd part, which is looking to the perspectives on RNAi solutions for plants. And for this talk, we invited Dr. Patrick Rudelsheim. Patrick is, at this moment, he's general manager in Perseus. He obtained his PhD in University of Antwerp in Belgium. And then he has a very rich curriculum where he started in plant genetic systems. And then also he moved on in Agrevo, Aventis, and then also Bayer. And now he's the general manager of Perseus. Patrick?
Yep. I hope you see the slides and you can hear me. Yeah. You can hear
Perfect.
me. Yeah, okay, perfect. Thank you very much. Thank you for the very kind words. I hope I will stand up to the expectations now. Thank you also to the Cost Action to invite me for this. I see it as a continuation of a meeting, webinar, no, not a webinar, a meeting, a real meeting. Imagine a year ago in Athens, where we discussed the communication aspects within the project. Not only this, I think it also, I want to thank the organizers, especially for bringing me after 25 years in the same session as Mark. Mark, you mentioned your history of destroying a field trial. I suppose actually those were our plans. So it's good to meet finally in this kind of environment where there's more room for discussion than for action. Okay, as he said, I'm with Perseus, we're a service company, but I will not go into that. I only have 5 minutes, and I will use the 5 minutes ideally to at least provoke you a little bit, because when we're talking about users' perspectives on RNAi solutions, this was the title I was given, obviously the question is which users and what kind of solutions do they need? And to picture this, I have this very simplistic scheme of a chain of different stakeholders, different parties that are involved in research, where you could be interested in just understanding how RNAi works, what it can do. So you're part of technology development, where you are looking at making it a product, which again has very different players. It could be the people who are interested in bringing the product and making finally something that works on a very specific crop for a very specific purpose, to production and distribution. We know that that's actually a kind of a business of its own. It has its own dynamic. Up to application, farmers will be using this. How do they integrate it? We've heard about the farmer's perspective of maybe better in-plant rather than applying it, but then again with all the complications of GMOs. But then after that, of course, it's not finished. We move into harvest and processing. We move to the retail sector, and eventually we come to this individual, the consumer. I'm showing this because in essence, they're all users. We're all users to some extent. the point is that obviously the real user in this scheme, the real user of the technology will be the farmer. And when we were talking about benefits, I like the point that also previous speakers stressed the point of make sure the benefits are understood. The benefits are essentially for the user, for the farmer. How do we translate that message to the rest of the chain? How do the processing industry, how does retail see these benefits? And can we convince the individual consumer of this? So what is important, I think, in this scheme for me is indeed to say it's clear that we have different players, very different type of players, all with their own interest, but also with their internal dynamics. And in each of these groups, you have changes, you have competition, you have people who try to position, organizations that try to position themselves for the scarce resources that are available. What's important also is that communication hardly is relevant beyond consecutive users. I think people may be interested in the next set of players, but they will not necessarily bridge. And I think part of the difficulty that we've seen with GMO, I think, is that people in basic research, people in technology development, suddenly had to bring a message to a consumer. They were not used to that. The consumer is too far downstream in this process. You will not hear, although you have computers which probably have a sticker on it, Intel inside, I'm sure that you never had an engineer from Intel telling you how great the technology is that they have been using, unless you are, of course, an IT geek, then maybe you're interested in that. But as an individual, you're not interested on what is inside your latest iPhone or latest smartphone. You will use it and you love the pictures that you see, but you have no idea how it's made. So why is it different in this case? And why do we think we're going to succeed? Mark was already indicating the problem of communicating about RNA and how people will remind what is there. So this is obviously a challenge. On the other hand, and I think this is then also the reason why I would agree with Mark on the not being too optimistic about the impact of the RNA vaccines on other RNA applications, is that it's a very different set. I think if you look at this scheme, you're seeing that the application of the product and the benefit of the product are immediately linked to that consumer. As a person who is going to be vaccinated, you're going to have the immediate benefit and you're going to have the immediate exposure to the product. Whereas if you're thinking about integrating it into a farming community, then again, we stretch that distance, and I'm sure that it's not going to be perceived in the same way, and that people will probably have already other ideas about use of RNA in plants. To make another analogy with something that is happening right at this moment, this is from an action that is ongoing in the UK at the moment. There is an action by Slow Food and a number of organizations to ask people to write to their supermarkets to post against gene-edited crops, gene-edited products. Again, this is, you could say, yes, this is another action. This is what they do. They have done this with GMOs. Remember that initially Flavor Saver tomato was put on the shelves in the UK supermarkets, but then was taken off the shelves because of these type of actions. We're seeing the repeat of this event now happening with gene-edited crops. It's going on, so we'll see how it works. Already the co-op has already indicated that they will not shelve it, but I just want to use this as, and particularly a quote of one of the activist chairpersons. They said, look, with co-op indicating that they will not shelve these gene-edited products, yeah, it's reassuring the majority of the UK public who don't want gene-edited foods on their shelves to show that their wishes are respected. And I'm certain that they will gain a competitive advantage over other stores. I think this is clear that it's linking those two last steps in that chain, linking the consumer. And I'm not going to discuss whether indeed she's right that this is the majority. I think you've seen the figures from that Mark has shown, then majority seems at least exaggerated. But still, it makes the link between that group of users, the last part in the chain, with the immediate previous link, the stores, and it's clear that this is about competitive advantage. Yeah? This is directly into the benefit or into the benefit of the interest of the retail stores. So if we continue to think that with our discussion on benefits for the farmer, we have anything that will influence these kind of actions, I think we're probably still on the wrong track. Science and technology, do we have a role in communication? Absolutely, but we also have to realize our reality. Our reality is that most scientists, most researchers are not comfortable with public communication. I dare to say that we are framed by correctness, yeah, and I put that between brackets. We're framed by facts, we're framed by statistics. Yeah, we know that there is never an absolute, so that makes us very, very tricky in communication. It's also clear that we have different objectives. Yeah, it's not always the same language. If you listen to gene editing or genetic modifications or RNAi, occasionally we tend to point to each other. Yeah. Many researchers, many scientists are bound by project timescales. Yeah, so long-term communication strategy is usually not in perspective. Messages can be vague. Yeah, they are not always very clear. There's also no clear voice, and we are not very well organized. Mark was referring to it's not necessary to make a consortium or to make like one big group, but it's very difficult if you are not organized. So key point here, and this is also what we discussed last year during the workshop, is that it's probably not necessary to commute more, because probably a lot of people say we should communicate even more. But I think at least let's try to spend the effort that we do in communication and try to improve understanding the different perspectives and trying to understand what people see as an important message. I would like to keep it to that and then listen to questions and see how we can conduct the discussion. Thank you.
Thank you, Patrick, for bringing here a lot of nuance and bringing the discussion, feeding the discussion. Thank you very much. I think the three papers really gave a good view. I also want to bring in here something within the new call from the European Commission, Farm to Fork. Also there, this Green Deal call by Ursula von der Leyen, where she also said that she wants to finalize her mandate with a GMO on the market. This also has put a lot of attention also in the public domain, and is really a mind changer as compared to 10 years ago, specifically for Europe. Another thing I want to bring up here in the discussion is that also at the European Academies, the Scientific Advisory Council also is working on this specifically for environment and agriculture. Of course, this is also a little bit in the public domain, but of course, this is not the Facebook and the Guardian. But I think there are also elements that feed the public perception. We know that communication is perception creation. With this, I think I look a little bit to the comments that are given in the chat here. Bruno, you made a comment on the talk of Mark. Of course, you will always have believers and non-believers. But your question is on the opposition that is not accepted, that it is assumed that GMO is associated with multinationals. How have you any ideas how to change this perception? Mark?
It's almost impossible to change a widely held public perception once it's become deeply rooted. It isn't always the case that a new technology will become demonized and opposed. I remember back when the GMO debate was playing out, a lot of us in the activist scene thought that nanotechnology would be the next big thing. And there was a lot of groups mobilizing to oppose nanotech. And that never really happened. It didn't catch, there were some attempts, but it didn't really catch the public imagination. And nanotechnology is out there and is pretty uncontroversial. Another example, though, from the other side might be nuclear power. So you would think, logically, in the age of climate change, that a very large source of zero carbon electricity would be something that even environmental groups would be able to have a serious think about. But there's not really been any shift at all, as far as I can tell, in the kind of pro and anti debate on nuclear. Even to the extent that countries like Germany, which are exiting from nuclear power, will continue to burn coal for 10 added years and emit millions of tons of additional CO2, and get support from the green groups in doing that, or at least to the extent that they would prefer not to have nuclear than to, even though they say that climate change is the greatest emergency ever to confront humanity. So there's no logic to this. The problem is that once these narratives are very firmly established in at least the minds of the active public, who can be very influential with everyone else, it's almost impossible to change it because the narrative is then so strong. So the storyline is with GM, as was mentioned, this is about big companies. It's done non-transparently. It's potentially unsafe in terms of food. It might have long-term effects. It might cause cancer. These are things that my kids are being taught by the teachers in the schools, by the way. So this shows how, like, just in my own personal experience, I find it shocking, but this just illustrates how widely held these views are. And all of that. And you can't, and even when, I mean, it's a bit like vaccines. I mean, it seems extraordinary that in the context of a pandemic, which has kept us all shut at home for a whole year pretty much, and, you know, killed 500,000 people in the US alone, and all the rest of it, that anti-vaccine sentiment should increase rather than decrease. Like, we all thought, those of us who have campaigned in favor of vaccines, that, well, if there was ever a pandemic, the anti-vaxxers would certainly disappear. But that's not been the case at all. So even when it's very clear that there are benefits to the use of a technology, if the misinformers can be successful on social media, and this is of course a time when conspiracy theories and anti-science and things are becoming increasingly evident, then you're not at all guaranteed a success. So I'm sorry to be kind of gloomy and depressing about it, but it is important not to be naive and to understand just how serious this challenge is. It's not just public communication. It's actually something much more, much deeper than that.
I think this was clear from from the three presentations, that first of all you have to make clear what is the benefit, and the investment of communication should be in that, and not to explain the technology, but really to present the benefit first. I think this was clear for the three presentations. And that is the driver to change mentality. And
very briefly, so but the question is not to convince everybody, but to convince at least the majority. At the end, we are going to vaccinate. So the question is, clearly some people will always remain in that position, will never change. But the issue is, are we able to reach at least the majority of people that will accept? And so to change, because that is the issue.
Yeah, my response is you don't need a majority. There's not going to be a public referendum on this. You need to have the elite opinion formers essentially convinced, and that means the political establishment and the mainstream media, if you like, because what's holding up any use of GMOs in Europe, it's not that the majority somehow gives a shit. It's because of the regulatory environment, which makes it impossible to ever deploy this technology, right? And that was established through a political process in Brussels ultimately, and that's why it's going to be so difficult to change, because then all of the different member states have a very vested interest in this and so on. But so you can be quite targeted, and I think it's very important that the scientific community is united on this and makes a very clear united position, which then makes it more difficult for the politicians to hide behind populist anti-science sentiments.
Just dropping also the idea, RNAi is not GMO. I mean, it's not at the level that you change the genetics, it's at the level of the production of a protein. So maybe that's also a misperception that has to be tackled out. Maybe
but is gene editing GMO?
No.
Like, would you say gene editing is GMO?
Yeah.
But that's
exactly. So it depends how the narrative is framed. That's my point. You see, it doesn't actually matter technically how this stuff works.
Mark, there was also a question for you in relation to what has made you changing your opinion. Also, Thomas Crampton asked for that. What made you changing your opinion? And if you can give any concrete example of what has worked to change the opinion in this context.
For me, it's a complicated and quite a long story, and for that reason, I wrote a whole book on it called Seeds of Science. So, chapters 3, 4, 5, 6, 7, and 8 basically explain how I changed my opinion. It's not something that happens overnight. I mean, for me, it was about becoming a science communicator in other areas, particularly on climate change, and understanding and communicating about the scientific consensus on climate in order to kind of to challenge the opposition of climate change deniers. And obviously, I found it ultimately that I couldn't go around shouting about how important scientific consensus was on, say, climate change and ignoring scientific consensus on the issue of GMOs. So for me, it was about being consistent as an advocate for science, if you like. So that was an unusual situation. It's because I'm a writer. It's not something that you'd expect everyone to go through. It's because I had to really spend a lot of time sifting through the evidence, and it took a long time to do that. But I don't, there are some signs of change. I mean, ultimately, the environmental movement is to blame here, and we need a better environmental movement, which is progressive and which is pro-science. And I'm involved in a set of efforts to build such a thing. We call it eco-modernism. We've now got eco-modernist groups in most of the European countries. And they support nuclear, they support GMOs, but they also support aggressive climate change mitigation. So you don't fall into the kind of classic left-right dichotomies here. It's progressive, it believes in growth, it believes in innovation, it believes in technology, but it also believes in saving the planet and protecting biodiversity and rewilding and stuff. So I think we have to kind of start at the grassroots as well in terms of building up a new social movement which supports science, but also has strong environmental objectives. And hopefully that will pay dividends somewhere further down the line as well.
Thank you. I think what we also should learn here from the session is that communication as scientists, as a scientific community is very important. But I think we should also invite journalists that can translate it and bring it to the wider public to change, because communication is not only perception, but it's creating perception. That is to some extent what I learned here from this session. So with this, I also invite everybody to follow the organization and to follow the webinar organized by Vera on the 4th of March. And then we go to the second session, which will switch from, let's say, RNAi in agriculture to more medical interest for RNAi technology. Hilde?
Thank you, Guy. I really enjoyed it. It's so nice to be together, even though we are not seeing the whole the whole picture. So now, as Guy said, we will more focus on a possible bridge, or at least mutual use of the technology in health and what that might mean for environment and food production. So I'm seeing this as a tour around Europe, and then we will get across to the Atlantic Ocean too at the end. I was really thrilled by a paper of this nice Sihan, but I don't know how to pronounce your name, Tastan. So I think I would just say that I really enjoyed your paper, and I'm so looking forward to your presentation. And if you could start by shortly introduce yourself with what you think is the most relevant for this audience, I would be very thankful. You have to unmute your microphone.
Can you hear me now?
Yes, thank you.
Okay, dear Dr. Hildegard, thank you very much for your nice introduction, and I am very pleasure to see that you see our paper and you pleasure to read that. And dear audiences, welcome again. And I am very excited to hear such an improvement and a discussion on the RNAi technology. So now I'm gonna present about the RNAi technology in the health medicine to be able to cure some diseases. Let me open my presentation. Okay, so can you see clearly?
Yes, very nice.
Yeah. Okay. I am Sihan Taştan, or in Turkish you can call Cihan Taştan. I'm director of the Transgenic Cellular Research and Development Center in Üsküdar University. So basically, we are working on several different genetic engineering tools to be able to cure the diseases, especially the rare disease or genetically disorders, and also the cancers. And in these technologies, RNAi keeps the very important stage to be able to find a new therapy tools. So today I'm going to talk about the diagnostic and the therapeutic applications of RNAi technology. So basically, all we love the flowers, and all we know very well that we discovered this RNAi biological mechanism in the transgenic plant Petunia, and now we are very well knowing that there are the very small nucleic acids that can downregulate gene expression in the plants as well as in the animals and the humans. So after discovery of the RNAi technology in the flower at 1990, then 15 years passed to be able to test and discover same mechanisms in the C. elegans and some mammalian cells. So after obtaining the Nobel Prize by the Dr. Fire and Mello, then the publication size just bumped after the couple of years. So in the human, we know that there are a couple of different mechanisms using these approaches to downregulate the gene expression, such as the small interference RNA, microRNA, or the we call it RISC as a RNA-induced silencing complex, and we have some enzymes, dicer and drosha, that can initiate RNAi downregulation mechanisms. So basically, all the mechanisms aims to downregulate gene expression in human cells. So why we choose or why we prefer the use of RNAi technology in human disease therapeutics? Because it have very good advantages to be able to downregulate genes. By the way, it is not a permanent gene therapy, so its toxicity and its dangers is very low. So it has high security and cost effective. And since it has high specificity, so we use this gene therapy method to be able to cure specific genes or to downregulate specific genes, as well as we can label it to be following, and it is very easy to transfer and to use. So basically, in diagnostic and therapeutic applications, now there are many different studies and clinical therapies on infectious diseases, stem cell biology, oncology, and hematology. So there are a couple of different approaches to be able to diagnose the disease progression, and as well as to therapeutic approaches to cure the diseases. So after the Nobel Prize, especially in the infectious diseases, as we know that there are bacterial infections as well as the viral infections, there are a couple of different RNAi usage to be able to use in bacterial infections as an alternative to the antibiotic, because antibiotics are not specific and it can cause the decrease in the or commensal microbiota. So that's why the RNAi technology is with its high sensitivity, we can use to downregulate bacterial infections, and as well as we have the HIV infections, West Nile virus infections, hepatitis C virus infections, and influenza. And these all different viruses are affecting human population very high, and thousands of people die in each year because of these infections. So since these viruses are evaluating and changing because of their mutations, so RNAi technology with recognition of specific gene or genome site in these viruses, they can downregulate specific genes by inhibiting of their replications. That's why now the RNAi technologies are using as an antiviral therapy agents. So in our paper recently published in Gene Therapy journal, we discussed the RNAi silencing methods as well as especially on the COVID-19 pandemic. So we know that SARS-CoV-2 viruses are RNA genome virus. So since the vaccine development is getting slower, so RNAi with their high specificity and usability, we discussed in this review paper that we can use RNAi as an antiviral agent in SARS-CoV-2 infections. So especially there are a couple of different studies, especially patents are taken for SARS-CoV-2, SARS-CoV and SARS-CoV-2 infections, and maybe in the future years we are gonna see that this type of drugs from RNAi can be used instead of the vaccines. So this is the new approach for the RNAi drug. So on the other hand, we can use the RNAi system as a diagnostic. So in the central nervous system and immune system, especially during the progression of the diseases, we can see that several different microRNA, which is sub mechanism of RNAi, and they can be downregulate or upregulate their expression, and which is highly affecting the differentiation of the cells, especially in immune cells. On the other hand, especially in the nervous systems on astrocytes and oligodendrocytes. So based on these upregulations and downregulations of the microRNAs, we can use these markers for diagnostic of the disease progression. So especially there are a couple of different therapeutical applications too. So these are still in the clinical trials. So a couple of them are already in the phase 1 studies, especially on the hepatitis B virus, solid tumors, and HIV infections. But when we look at the other companies that are already in the phase 2 and phase 3, which are also focused on the age-related macular degeneration and diabetes, and on the other hand, there are a couple of the other diseases also. So what we know that in the closing years, we will have a couple of approved RNA medicines in macular degeneration and some viral infections. That's why, because of this couple of the advantages, as I mentioned earlier, this technology has a wide application. So thank you very much to listen to me, and if you can have a question, I can try to answer it.
Thanks a lot. I really enjoy that. It's and I'm not sure, we didn't really put up time for questions after each presentation, but if there are some, then I should look at the chat.
Sure.
I think we will take the discussion after the three speakers. So then that leaves people time to think if they have specific questions or matters.
Yep, my pleasure. Okay.
Thank you, Sian. I am not trying the other way of pronouncing your name, but maybe one day. So then, even though it's busy to leave Istanbul, we are going to travel to Zurich, to Hans Peter. Many of us know him from EFSA. Today he's going to talk as free or scientist with all his experiences. And he's going to discuss whether we could have regulatory effects of RNAi in agriculture due to the new COVID-19 world situation. So I'm really looking forward to hear your thoughts today, Hans Peter.
Yes, thank you very much, Hilde. Hi, Hilde. Good afternoon, everybody. I was asked to address this question that now that everybody knows about RNA vaccines, whether this can influence the view on agricultural uses of RNAi and also in the food production. Is my screen shared, Hilde?
Yes.
Thank you very much. So, I mean, with this, to answer these questions, there are two issues that spontaneously come to my mind. One is hazard perception in the general population, which has already been mentioned is very difficult to influence. And this is a social psychological issue or social cultural issue, of course, way beyond my competencies. And the second critical issue is how to communicate the outcome of a scientific risk assessment. That's also very difficult, not only for me, but I guess for many scientists. Once that the scientific risk assessment has been concluded, how to communicate to lay people the outcome of this assessment. So I will, this is, as I said, very difficult also for me, and I will come back to these two issues in a minute, just by giving my personal perspective, or try to give my personal perspective. So the issue of the fact that now many people know about RNA vaccines, know the word RNA, can this have favorable effects on hazard perception? And there are two sides of the coin, in my view. One is that there might be a favorable influence because people now recognize, particularly, of course, with the vaccine, RNA-based vaccine, that RNA is a substance or something that can be useful, that humans can take advantage for their own uses. So it's something potentially useful. So here, thumbs up. But on the other hand, the fact that RNA vaccines work also shows that RNA can have potent biological effects. I think people understand that to act as a vaccine, RNA must induce the production of an antigen. And an antigen can also become an allergen, which is a critical issue in connection with food. And also extending this thought, I mean, people could also realize that RNA may also lead to the synthesis of potentially toxic peptides or proteins. So here I see potential negative effects on, or less favorable effects on perception of people's perception. So here, thumbs down. And I think I share this view with the speakers that I heard this before this afternoon. So I think I come to a very specific example, very shortly. This example of an RNAi maize, which is protected against an insect pest. This maize expresses a double-stranded RNA. So it's a GM maize that expresses double-stranded RNA, which is then processed to a small interfering RNA that targets an essential gene of the corn rootworm, which is a common pest of maize. And you see down here that this siRNA leads to stunting and lethality of the insect when it feeds on the roots. So this RNA principle clearly protects the roots. You see here conventional maize, which has been heavily attacked by the pest, and here an RNAi protected maize, GM maize, which is protected from the pest. And I'm showing this example because now in my perspective as chair of the GMO panel, this is a milestone. The risk assessment of this maize has been a milestone because the question came up now, if RNAi can kill insects, is it also possible that it could be harmful to animals and humans? Or in other words, how is it possible that an RNAi kills insects but is harmless to animals, farm animals, for example, and humans? And here there is, I'm not going into the details of the risk assessment. There is an appendix that you can read summarizing the risk assessment, but again, now I'm coming to this issue of hazard and risk communication, because on the one hand, it's impossible to completely exclude a hazard to animals and humans. This is because of the biology of RNA interference in animals, in mammals and humans. So the biology is such that it's not possible to exclude potential effects just based on the sequence, just based on bioinformatic in silico analysis. So even scientists cannot completely exclude a potential hazard. But on the other hand, the full risk assessment came to the conclusion that it is highly unlikely, or even I would say extremely unlikely, that RNA and its derived small interfering RNA newly expressed in plants are able to exert any biological effects in animals and humans because they don't reach target cells. There is no uptake into target cells. There is no systemic uptake and so on. So because of these critical issues regarding hazards and risk communication, so hazard identification and risk communication and the problem of perception, I think there are two answers to your question, Hilde. Yes, I believe that the existence of RNA vaccines and the success of RNA vaccines will influence the perception, the public lay people perception, but it will do so in a negative effects for products that provide no direct benefit to the consumer. I think here I completely agree with what has been said this afternoon. I'm also very skeptical here. So as long as there is also only an agricultural economic advantage, the consumer acceptance will not increase. This can change, however, if there is a direct benefit to the consumer. This may favorably affect influence hazard perception in lay people if, like with vaccines, they provide the products provide a direct advantage to the consumer. So here there is a chance to increase acceptance among consumers with very clever products. And I think that's all I could contribute to the discussion of this afternoon. And my appendix just summarizes, for those who are interested, summarizes the full risk assessment of the example I made before. Thank you very much for your attention. I give back to you, Hilde.
Thank you, Hans-Peter. And it's really nice to get that appendix too for us afterwards. Yes, as you say, it's not easy to communicate the risk, and it's difficult to know what to compare it to. I hope you will stay on so that we can put you into the general discussion after the next speaker. So then we will travel from Zurich to Oslo, so back to Norway, for another economist. Sometimes it's, I don't know, I'm a biologist, obviously, but sometimes it seems to be easier to be an economist, where you can relate to figures and clearer or easier answers, maybe. Joachim, are you? Yeah, that looks very promising. Very good. So Joachim, he's a professor at the same university as I am, and he has been to several in Europe, so he knows this environment, to put it that way, well, for all over Europe. So I'm really looking forward to your presentation, Joachim.
Thank you so much, Hildegun. Can everybody see my slides and hear me? Can you hear me? Hildegun? Okay, very good. Yeah, all right. Thanks very much for inviting me. So you called me an economist. I do many things. I also do like risk modeling and so on. So I have hobbies. And nowadays I'm professor of innovation. I still also have like one leg in Zurich, so I still have affiliation with the Department of Informatics there. But innovation is my main topic, and in a way, the issue that has always most fascinated me is resistance to innovation. And I've been researching resistance to all things related to gene technology since, I think, 1996. So even wrote a PhD about that a long time ago. And when we talk about RNAi, then for me, the question is, all right, so is this actually a new thing, and is this going to be a big thing? So will RNAi become an object of public controversy? Now, when you take a look at Google Trends, this is actually quite a good indicator of how much people care and argue and so on about any type of issue. Then what you can see in the graph, it only goes back to 2004 because Google Trends is a relatively new technology, newer than RNAi. So we can see is that this is basically a non-issue, right? So comparing it to a long-standing issue of public controversy like GMOs, or let's say the products that are related to GMO technologies, such as Roundup, there has never really been much of a public interest in RNAi. So this is pretty much something that is discussed only in our circles. So scientists, regulators, and so on. The public hasn't really caught on yet. Now, the question is, of course, will it do so? And when we think back about what it was like in the late 90s, when we had these big wars about GMOs, and much of this relates back to Mark's presentation from the very beginning, then this did not happen all of a sudden either. So GMOs as a, let's say, PR war zone, they were developed. And now we've also discussed other types of technologies that might have become a big issue or have become an issue at some point, but are not anymore. So things like nuclear power, things like nanotechnology applications, etc. And when you try to look at the career of these controversial technologies, then we can try to distill certain conclusions that are maybe not based in hard science, but they always sum up pretty much what happens and what can happen and must happen before a technology or any other issue becomes a major public controversy. And this is my take on it. So what does it take to raise a big controversy? So I think, so lesson one that can be distilled from all our experience with this in the past is that the technology must, in public perception, be linked to some latent threat potential that can also be vividly communicated. If it cannot be communicated, it cannot be made into an issue, right? Then in order to be, let's say, a real public controversy, you also need to know what is being threatened here. And when you look back historically, then technologies that became major public controversies, they were often perceived to be in conflict or threatening widely shared value orientations in the general public, and also value orientations shared by stakeholder groups. Then the third point is maybe the most important one for us today. So an issue does not become an issue unless a major player in the issues management world decides to take it on and develop it. And if you know, for example, people who work at Greenpeace, so Mark, you mentioned a couple of the old gang there in the beginning, then think about how Greenpeace does issues management. Greenpeace treats issues, campaigns, like a CMO treats a brand portfolio. These issues are built up over a long time. They are fit to each other, so they are actively managed. An issues portfolio for a major NGO is something that is like a political program. It is also something that is related to a business strategy, because it helps if there is some potential for cashing in on this. So these might be, let's say, negotiation assets that you can use in political horse trading. It might be issues related to fundraising possibilities. It might be recognition in the public. So an NGO has to gain something from an issue. If it cannot gain from an issue, then there will typically not be anything happening. Another thing that experience suggests is there must be some sort of trigger event, either occurring spontaneously or engineered, that creates viral coverage. And yeah, in the beginning, Mark showed a couple of these that were responsible for what happened in the late 90s. So Dolly, the field destructions of the farm scale trials, the Butterfinger thing with Nestle, and so on and so on. So without that, it's almost impossible to develop the momentum that makes an issue a public controversy. And the momentum must also be kept alive. So this typically doesn't happen unless the debate creates sufficient polarization and sustainable polarization. If that is not the case, the whole thing is over very fast. Now, these are sort of my subjective words of wisdom based on what I see as what all the past issues with controversial technologies have in common. Now, the question is now for us to discuss if RNAi actually has what it takes to become a major public controversy. My personal opinion is no, it doesn't. There are some of these elements. So the first two, one might argue there may be something. The rest, way below threshold value. So I don't think RNAi will ever become a major public outrage issue. But that's just my belief. So let's discuss. Thank you so much. And back to you, Hildegunn, for the discussion.
Thank you, Joachim. We are almost neighbors today, I think. I see you're at home too.
Actually, today I'm the first time in the office since.
Oh no, that is office plants. You're right. You have a nicer office than
plants
Nice.
haven't really survived much. So yeah, we'll see.
It's not much down regulation yet. I really enjoyed your presentation and all three presentations. So the question is where to start. Maybe it's easy to kick it off by your what I would say positive perception that RNAi doesn't have what it takes. I think I would be happy that it doesn't have what it takes. Maybe because it's complicated and it's a lot of different products, so it's kind of difficult to put it in one bag. But but do you have an issue, or can you explain why suddenly genome editing became or had what it took? Because in the beginning, most people thought that would be different from GMOs because it wasn't big industry necessarily, and it was a very surprisingly positive start. Do you have an idea or opinion when it changed?
Yeah, my opinion is that it never actually changed because there was not really any public controversy about this. It wasn't a political issue in a way. So what the regulators said is that as the regulations are now, you will also have to treat genome edited plant products under those same regulations. So you could argue that the Court of Justice just passed the buck back to the legislators and said, okay, you know, if you make shitty laws, we have to go by your shitty laws. In a way that is, so as I understand this deep down, yeah, the court says, hey, you know, politicians get together, think about this again, then we can also say, you're right, genome editing is a different issue. But with things being as they are, that needs to be regulated under the existing regulations. So, but in terms of public controversy, I don't think anything ever really happened. So no major scandals, no front page news on the negative side. So I don't think this was ever really an issue.
Thank you. There is a comment here from Sina Ben Ali saying, what about presenting the RNAi technology with detailed explanation in public television program as well as in social media channels? And after explaining the technology, scientists can be interviewed. And what about interviewing anti-GMO activists? The problem there, the way I see it, is that we wouldn't like RNAi to be directly linked to GMO, just as GE to us isn't a direct link to GMOs. So that, I guess, when we talk about benefits, that is also a channel or a challenge when we can't really compare it to today's standard, because then we say something wrong or we link it to something we don't want to link it to. So I think that issue is really difficult. It's what do you think, Joachim?
I don't know. So you could ask yourself, why do we want to create some positive sentiment in the public about technology? People in general don't give a shit about technology. People are interested in products, right? Technology that has such widespread applications as RNAi has, is almost impossible to sort of develop a concept that is somehow tangible. So I suppose, why should we actually make an effort? You know, the technology as such is not an issue in the general public. If we start proactively trying to develop benefit communication directed at the general public and so on, so why should we bother? We might think about, all right, why don't we let sleeping dogs lie? So de facto, there is no resistance, right, in the general public. And in a way, sort of what we need in order to continue on the technology development path is simply the absence of any resistance. We don't have to have active acceptance. So this is easily confused, but acceptance and resistance don't really have anything to do with each other. So so I wonder, sort of, is there actually a point in trying to educate the public about RNAi? I would say not really, right? So, I mean, people won't listen anyway. So by and large, and those that listen, they actively search for such information. And most of those that do listen are scientists anyway, I suppose. So us here sitting in this webinar. So I'm not entirely sure if benefit communication is necessary. So this is probably more something in stakeholder communication. So so if politicians have to make risk-benefit trade-offs in legal developments or regulators and regulatory decisions, fine. But why make it in a way a public education effort? Can't see the point.
That's a good point, actually. What do you think, Sian? I guess one of the differences we see is that people, how to put it, in the beginning we think that food is something essential we need every day, while medication is something we need when we have a problem. But it should be less, we don't need it as often as we need food, but still when people are ill or when they need a treatment, then it seems like they will take anything. So, and when you treat people, do they really, are they interested in how they are treated, or are they more interested in being well?
Yeah, to be honest, let's talk about the for vaccination, because in nowadays with this pandemic of COVID-19, so especially one vaccine is become very famous worldwide. So it is the mRNA vaccine. So based on this vaccine, just your immune system just react to the protein from the SARS-CoV-2. And in this vaccine, there is only RNA sequence. So with the help of this vaccination, especially population become familiar to get such a synthetic RNA or synthetic nucleic acid in their body. So they decrease their frightening. So in the next year, so especially for gene therapy or genetic treatments, so RNA applications and RNA interference technologies will become more acceptable to be able to approve as a medicine. So for one side, I am very thankful for this pandemic, because with this pandemic, these technologies are understood very well by the whole population in the world. So all the health regulations are trying to understand new technologies, and in next years, they will understand that these technologies are not permanent. Genetic engineering. So that's why I'm believing that they will accept these RNA technologies to use in the human body as a medicine. So that's why, especially when we make a classification in gene therapies, RNAi technologies, antisense oligonucleotide technologies, this type of technologies will be the first class of medicines, because it doesn't affect your genome. It just affects your second line of gene expression. That's why it is more acceptable as a medicine.
Mm-hmm. And that's a nice bridge over to you again, Hans-Peter. So if it should be so that people are not that concerned about RNAi or food made from that technology. How do you think, Hans-Peter, the regulatory might adapt? Could you imagine that the politicians or the EU Commission don't see that they would lose so much from accepting that kind of products, and therefore it would be less?
Yeah, to our country's regulations, they were asking such questions of what is RNA, or what are these techniques, and how it can be used in the plant also. So in my previous year presentations to the regulation committee, so I mentioned that these technologies are much more helpful for engineering or something engineering stage. So that's why in our regulation committee, they start to understand that instead of permanent engineering, this type of the regulation in the plant and as well as in the other medicine-like stages, they will try to look at positive.
Positive is good. Thank you, Siam.
Yeah.
Hans-Peter, what do you think? I saw you were flashing up your video.
Sorry, my connection is a little bit unstable at the moment. I hope that you can understand me.
I hear you.
Good. Yeah, I mean, the problem is that as long as there is insertion of new sequences into the brand, then it will fall under the GMO regulation, and that of course scares the consumer. That's clear. So I don't know. I mean, maybe there is some clever solutions for certain applications where this could be done by gene editing, exploiting endogenous sequences. I don't know if that's possible. But then we have the gene editing regulation problem in Europe. But as long as it is GMO, meaning that we open the chromosomal DNA and repair it by adding new sequences, then this will fall under the GMO regulation, and that's a problem, of course. So the question is whether the RNAi the RNAi technique can be used otherwise by spraying without the use of GMO plants.
Yes, so if it would be put on nano clay, for instance, how would
Exactly.
EFSA and how would you treat that?
Well, I mean, then it depends what will be the remit. So which this could then be considered a pesticide. And then it's certainly not anymore under the GMO remit, and you know, the regulation of pesticides is slightly different from the regulation of GMOs because member state countries are closer to the pesticide regulation. So yes, it could have a different fate, of course. Yeah. Particularly if it's something that does not persist in the environment. I mean, it needs certain persistence, of course, to act on the plant, to protect the plant, or to do whatever it should do, but then it should not persist, or it should persist less than current chemical pesticides, for example.
And could it be then classified as a biological control?
I don't know. I mean, that really depends on. No, I cannot answer that question. How it will be? I don't know the regulation in Europe. But from the people's perception, if you ask me, from the lay people perception, I don't believe so. I believe it's a chemical. It will be considered as a chemical, and could be classified basically as an agrochemical, hopefully.
So then the vaccines are chemicals too?
Well, I mean, that's sorry, that's a discussion that I always have. You know, I mean, our food is composed of chemicals. I mean, DNA is a chemical, RNA is a chemical, proteins are a chemical, but this is again is a question of perception. You know, chemicals are normally something which is foreign, which does not belong culturally, traditionally to that food, and then it's considered a chemical. But of course, our food is full of RNA. I think one of the points the panel makes regarding the assessment of RNAi in plants is that this newly introduced non-coding RNA is only a very, very minor fraction of the total non-coding RNA expressed in the plant physiologically. And that again is a very, very minute fraction of the total RNA which is present in a plant product. So we are talking about nanograms or less than a fraction of nanograms compared to milligrams of RNA. And that's but it's, as long as it's foreign, it's a new sequence, then it will be considered GMO. That's our problem in terms of risk communication then.
It's another interesting suggestion here from, I'm not sure how to, Bridie Maxwell, that she's suggesting to make a sticker, like a butterfly, non-GMO type of sticker that has a science basis for real environmental footprint, so the consumer can feel good about science-backed consumption. What do you think about that? That's a bit onto my mention of the Impossible Burger, where, for instance, in California, where mostly the attitude was against GMOs when the Impossible Burger came, and explaining why they could only make it by gene technology and replacing a meat burger with something that tasted the same. That really took the market and changed the attitude hugely to GMOs in places in the US where they had been skeptical to start with. So do you think one could make a kind of brand that would be environmental friendly or science-based solutions or anything like that? Anyone? Maybe Mark, Linus, what do you think about branding?
Yeah, actually, I think sorry, I'm relaxing a bit more here. Actually, I think the Impossible Burger analogy is a really good one. I don't think it's, and it illustrates the points that's been made by several speakers, that you have a clear consumer benefit, which is that you have a hamburger which tastes almost as good as the meat one. And even vegans have tasted meat. So basically this satisfies everybody. And in fact, the Impossible Burger company, I mean, I've interacted with them a bit. They had a very proactive PR strategy. They even made, so it's not just the heme, the kind of hemoglobin, the red stuff, which was produced by GMO yeast, I think, but also they were making a benefit out of using GM soya because they said it was more environmentally friendly with no till and stuff like that. So they really went all out to make the case to the consumer. And my experience, like I was in Cornell, and the line in the cafe for the Impossible Burger was out the door. People really love it. And I mean, the products are amazing, and everybody knows that reducing livestock is critically important for, you know, biodiversity and climate change objectives and so on. So it ticks a lot of boxes, and it kind of splits the environmental movement because everyone wants to reduce meat, but at the same time, a lot of the original environmental groups are still against it because it's GMO, like Friends of the Earth was opposing it, and so on. But it makes them look old-fashioned and stupid, and I don't think they'll succeed with that campaign. So the Impossible Burger is a really good example of how to do it right.
Yeah, I really love that too. I have a son living in San Francisco, and I couldn't believe it. It was really an exciting story. So what if, back to you, Hans-Peter, I don't know, does EFSA risk evaluate the Impossible food or any of it, or is it nobody in Europe that has taken the challenge yet? Do you hear me, Hans-Peter?
Yes, I missed your question. I hear you now again, but I missed your question.
The Impossible Burger or any Impossible food, is any of that risk evaluated by EFSA?
Okay, we do have not yet. We have in the pipeline. We have. I mean, of course, you know, we would not risk assess the burger. We risk assess, of course, the plant product which is used to produce then the burger. We do have something in the pipeline. It's a protein, a plant protein, which would be expressed in an, I don't know which kind of expression system, which then, you know, provides a taste like meat.
Yes, I think Hugh knows more about it. He talked about it in Athens last year when we had.
I mean, it's not myoglobin, because myoglobin would be the animal protein, but it's a plant protein which has similar properties also in terms of taste. So that's in the pipeline, and that's a GMO, and that will be assessed very soon. Probably this year, the opinion will be published on that.
That's exciting. So when you say very soon, is it this year?
Oh, I cannot make any promises if it's this year, but we are trying to accelerate. I mean, EFSA is trying to accelerate the risk assessment. There is this green program at EFSA, and so I'm very much supporting this. And the GMO panel, the existing GMO panel, is also you know, all members of the panel are really pushing to get to conclusions as fast as possible. I mean,
great. That was very exciting news.
That has changed a lot in the last few years, particularly with the panel. And you know, I mean, if I may say, because of the change of the regulation, food regulation in Europe, the existing panel now we finish its 3-year term, but we stay another 3 years. That's a very exceptional solution to allow the transition into the new food regulation. So we can expect, you know, all the mechanisms work, all panel members know each other, and there is very good collaboration with the unit, so we can expect that the handling of applications will be further accelerated.
That's really exciting, and that's efficient, because then you are well trained, and also then to get the transition to the new, that's brilliant.
It's a very exceptional situation.
Yes. Very nice.
Hilde,
Yes.
sorry, I think that we need to go on with the program.
Yeah. Okay, Bruno.
To different questions. Let's maybe have a final section of discussion.
Yes. We will have that too. Thank you, Bruno. And to me, it's difficult to communicate and talk and listen and read these chats. So I think next time I'm going to be a chair, I need one chat reader and one speaker, I think, because they are like me, they can't do it just in one sentence, it's in many lines. But I really enjoy it. It's good to see that people are excited about this. So then I think we will go via Hugh and then probably back to Norway again. What do you say, Hugh?
Yeah, hello. So thank you for the very interesting discussion so far. The protein that Hans-Peter was talking about is a plant analog of mammalian hemoglobin. It's called leg hemoglobin. It comes from a legume originally. But anyway, that was another point. So this is the start of session 3, the session called Communicating RNAi. We have 3 speakers, and I will kick the session off with Joe Royslien. I'm sorry about my Norwegian pronunciation, Joe. He has an academic position. He's a filmmaker, and he's also the director of a science communication company. And I believe you're going to speak a little bit and show a film, Joe.
Yes, indeed. I'll try to share my screen here so that everything works out nicely. Share sound. There we go. If I do like this, you can all now see my presentation beautifully.
Yeah.
Perfect. So thank you. My name is Joe. So as he said, I am a mathematician. I'm a professor of medical statistics, so I have an academic career on the one hand, but I've also been working extensively with science communication for the last decade as a writer and as a host and executive producer for various science TV series on Discovery Channel and Nordic national broadcasters like Danish Radio, NRK, and stuff like that. So I care a lot about science communication. And for all of us who care about the communication of scientific knowledge, the COVID-19 pandemic has really learned us a lot. Because on a press conference on March 12th, 2020 in Norway, Norway was basically shut down by the Norwegian government. But what's striking to me about this image from that press conference is who's talking? Because it's not the Prime Minister, and it's not the Minister of Health and Care Services, it's the Director General of the Norwegian Institute of Public Health. So for months now, Norway, along with a lot of other countries, have to a large extent been run based heavily on the advice from scientific communities. And if science all of a sudden now is truly the backbone for running our country, then people need to understand what it's all about. And we, the scientists, we need to talk in a way so that our message gets across to politicians and to the general public. But how do we do that? How do we talk so that people understand what we're trying to say? And spring 2020, the Research Council of Norway, they had an emergency call for COVID-19 related research projects. And we were awarded funding for a project called COVCOM, COVID Communication, fighting a pandemic through translating science. And the whole point of that research project was to explore science communication issues related to pandemics, like washing your hands. And we know that washing your hands is important. And there are different ways of communicating that. One alternative is to just shout at the top of our lungs. We go, wash your hands, right? That might work. Maybe you would wash your hands because somebody told you to. Or maybe you wouldn't, because you don't understand why you're supposed to wash your hands, and not everyone likes to be told what they're supposed to do. So we could try other strategies. For example, I could tell people that this particular virus, it is encapsulated within a fatty membrane, which is great for the virus because it means that it dissolves immediately as it enters the body. But also, have you seen what happens when you wash dirty dishes in the sink and you add soap? Well, all the fat and the grease just goes away, because soap is awesome at taking care of fat. It kills the fat. Boof. And if I tell you this, maybe I don't even have to tell you that you should wash your hands, because you get it, right? You understand, oh, the fatty membrane and the soap, it will kill the virus. Oh, that's awesome. So this explanation, it will take longer than just shouting, wash your hands, but maybe the end result will be better. So things like that is what we wanted to explore in this research project. So we put together a lot of very different people. We put together risk communicators, public health communicators, media communicators, and then we start discussing and debating how to reach out with scientific knowledge and pandemics. And after a couple of months, we realized that we were not talking about the same thing. Because if you talk to risk communicators, to them, trust is key. If you don't have trust, people won't listen to you, and it doesn't matter what you have to say. And the importance of trust with respect to COVID-19 and pandemics has been established in multiple scientific studies. And we know that healthcare professionals are viewed as credible sources, but that trust in governmental resources and politicians has been a lot more unstable. Funnily though, research shows that while people tend to trust healthcare professionals more than politicians, when we put healthcare professionals on screen to talk, we tend to dress them like politicians, and then their credibility just crunches. Actually, a content analysis of Canadian TV clips show that a reason why Canada lost control of their message in mass media was basically due to the choice of spokesperson in press conferences, because their spokespersons looked like bureaucrats and they lost credibility of their message. So in risk communication, trust is key. But in health communication, the story is different. That is all about literacy. We want to teach people something, learn them something. We want to give people information so that they can make good, well-informed choices about themselves and their own health and the people around them. And research tells us that people report greater knowledge when viewing non-narrative videos as compared to narrative videos about facts and science. Actually, in the study, videos that were aimed at just eliciting feelings of anticipated regret because you never vaccinated. Films like that were viewed as patronizing and unprofessional. They were just too focused on the emotions and the regrets, and it didn't work out at all. That was an experimental study where they really tested this out against one another. Yet, while being factual seems to be a plus, people learn more from it. The use of narratives and messages that elicit emotions create more engagement. People choose to watch that instead. So it basically leads us to the media landscape, the place where the communication takes place, where everyone's fighting for everyone's attention all the time. And in media communication, it's about the catchy and the attention grabbing, because if it doesn't grab your attention, well, people will not click on it. And if no one's watching, it doesn't care what you have to say. And the WHO even has guidelines regarding this, stating that engaging in effective communication should be multimodal, across different platforms and different sources of communication, and it should include visual information. And the reason for the latter thing is that we know from a lot of quantitative research that messages that includes video are retransmitted 63% more than those without video. Videos fly across the internet, and if you cut that out, well, it won't work that well. And in a study on COVID-19 posts on social media from the Chinese government show that they had really low engagement with what they posted. And exactly because of that, because they didn't use rich media properly, and they used non-personal and non-narrative content. We know people learn more from it, but they don't engage with it. Also, we know that posts on social media with positive emotions create more engagement than negative emotions. So a project that tries to communicate knowledge and science sort of places itself in the cross-section of very different fields with different focus. And for us with COVID-19 and pandemics, we had trust for risk communication. In health communication, it's about literacy. We want to increase people's literacy, what we're talking about, so that they can make more informed choices. Well, in media communication, the focus is on the attention grabbing and the catchy. It doesn't matter what you have to say if no one's listening. So in order for a good communication to work, all of those three things has to be balanced. So the brief summary of what I said is mine now is really poorly chosen acronym THONK. I understand right off the bat from a communication perspective, I need to work on that acronym, but THONK is the best I have for now. I'll just develop that later on. The summary. If you want to communicate something, it has to be trustworthy, but it can't be boring because people will not watch it. They will not interact with it. It has to stir some human emotion. We're humans after all. We feel stuff, yet it cannot be the emotion that itself that's the key element. It cannot be too emotional. It has to be optimistic rather than pessimistic in order for people to engage with it. But still, it has to be realistic. We can't lie to people. We know that people get more engaged with narratives and stories, yet they learn more from actual facts and information. So we can't stray too far off the non-narrative towards the narrative. And finding that balance can be a true struggle. And then it needs to stand out. It has to be creative. It doesn't grab people's attention. People just keep scrolling down their Facebook feeds and they will never click on your post. And what you have to say will just get lost in everything. So in order to be truly successful, all of this needs to be balanced. That's what science says. This is based on review studies of the literature. And we are scientists. We should listen to science. This is what science tells us. So I'll end this brief talk about what we've learned from COVID-19 communication with a brief example. It's a really recent one, and it's from the Norwegian Institute of Public Health. And they had a professional ad agency create communication for them about vaccines and created a film for them. So it's an infomercial aimed at the Norwegian population, and it's about the distribution of vaccines. So remember, trustworthy, but not boring. Emotions, but not too emotional. Optimistic rather than pessimistic, but still realistic. Create a narrative to engage, but keep it factual and informative. And then be different, be creative, but not too crazy. So let's see how they did.
We are finally getting closer to normality. We have stayed at home a lot and kept social distancing. No traveling, no parties, no visits, not even a hug. Finally, we have a vaccine, and priority is being given to those who need it most. The eldest, the weak, and healthcare professionals. Unfortunately, there is still a long way to go, and many have to wait their turn. But this is the beginning of the way out of the bubble.
Yeah, so what do we think? I mean, from a scientific point of view, did they succeed in balancing it all? Was it a good attempt? Honestly, I think they did really, really well, because it's a hard struggle, and I think they balanced it well. And COVID-19 has been a huge learning experience for us who do science communication across the globe, within a whole range of different fields, because that huge amount of science that needs to be communicated really fast has sort of escalated the learning effect for us. And it turns out, unfortunately, that communicating these things to the public is actually way harder than we actually thought. But the good thing is that the scientific community has now started research into science communication and started slowly to untangle why this communication is so hard. And once we know why, well, then it's way easier to create a solution. So thank you.
Thanks, Joe. That was really thought-provoking, and I'll remember thonk. Let's leave the discussion until the end. We're a bit behind time, so let's move on and get the second two, the last two talks through, and then we can open the floor for discussion, including your work. So next up, we've got Hildegard Stoltenberg, who I know well from GMO work and generally socially. So she works at NULS in Oslo and is a professor of plant development and also a working group leader on this cost action. So Hildegard, the floor is yours.
Thank you, Hugh. Do you see my presentation now?
Not yet.
Because now okay, share maybe. Does that
Yeah.
work better? Good.
Yeah.
I think mine will be short, and I really loved the way Joe did his part. It was great. I saw it last night, and I got really happy. Okay, good. Here I'm just again getting back to this YouTube short video that is showing the history of RNAi, where they have it's like monks copying the DNA into recipes for the chef to cook proteins, and then if there are any fishy secrets or something looking like a virus, then the cup is there to protect, or the guard or whatever, to protect the cells and then get rid of these invaders. So I recommend that video. I'm not going to spend time on it now, but I really love that one. So, so how do I shift? Okay, good. And then as Mark and I just discussed the impossible food, I think the way this was a biochemist at Stanford University that realized it wasn't enough to communicate or talk. So he started this company they started off by saying, we don't need to get to the vegetarians because they are already eating well, but we have to help the meat eating 95% of the world population. And then they asked, what is tasting meat? And could we add that to a plant base? And that's where they did what Hugh and Hans-Peter already talked about, getting production of leg hemoglobin, this blood soup, if you like, that they add to the plant base. And to avoid having too much soil erosion and all of that, they put it into an efficient bioreactor, and that happens to be a single-celled organism. So in that sense, it's GMO content, or the leg hemoglobin comes from a GMO organism producing it. But this has really hit the market in the US. It saved Burger King. They got people using them again. The stores that got the product some years later, when they were able to produce enough, it became the most selling product in these stores, many times more than any of the other products. So that to us was really good. And they, in their communication, they talked about food for thought. They talked about, where's the beef? There is none. And just the use of the impossible word to me was really brilliant. So I've been reading their pages just for the fun of the wording. And then we have other people that have other ways of communicating than scientists, using different wording and getting results. So the question is, and this is the bridge over to Greg, we talked about it in Athens in this cost action meeting last year, and we've also talked about it earlier. It's the difficulty sometimes of choosing the right words and what that means. And then, of course, if we don't have to communicate so much as we think, or maybe we should have another goal with the communication. But I believe that, at least in Norway, journalists stopped using the word manipulate when it came to GMOs. And for RNAi, we could talk about solutions instead. And some would also say that gene knock-in or knock-out doesn't give a good association. So maybe genetic fine-tuning, as Greg will get back to too. And mutant, I don't know. Now with this COVID, maybe mutant has changed, but it's still something that is scary. So variant would be better, I think. Risk is also probably depending on culture, as we have said several times today. But maybe talk about opportunity instead, because at least in Europe, people don't like risk. Technology, as many of you said, it's not really the technology maybe that is what we choose, but it is the tools or the point of it. So basically the solutions. So it would be better. And also for biology, it seems like technology, when it comes to a tractor and using GPS, that's really something all farmers and people embrace. But when it comes to biology, it seems like technology should be far away. And that to me is a weird link. And then control might not be good for many either. So maybe help and protect would be better phrases for RNAi. And I think that's all for me. So then I send it over to the UK again, Hugh.
Thank you, Hildegard. And can I just remind the audience, anybody, please contribute to the chat, because we will need some topics for discussion. So if you have questions, comments, or you want anybody to expand on any of their ideas that they've articulated, then please use the chat box. So the third speaker we have this afternoon is Greg, or it's probably morning still for him, I'm not sure. Greg Watson from Bayer Crop Science. He's an expert in regulatory policy for crop protection. Over to you, Greg.
Thank you, Hugh. Well, I will try to get myself going here. Thank you for the opportunity to be with you this morning, this afternoon. And what I've been charged with is trying to provide an overview. I'd actually used American. I changed the title, took the advantage of doing that. This is more of an industry perception that I'll try to present today in terms of the vocabulary that we should use. And what I will share with you is based on a professional communication consultant survey that actually was done in the US as well as Canada and Europe. And we actually tried to start with communication to stakeholders that we normally work with in the agricultural space. Growers and other agribusinesses, the value chain, which connection to connections to food companies, as well as science communicators. And so there were 24 individual in-depth interviews, and we basically tested some of the word messages. And to give you some insight into that, we really were focused on a quest to what's the right public messaging. And my interest in this is, as a person that works with regulators, it's pretty obvious, right? Regulators are people too, so the language that you use to communicate when you're working through the regulatory process is really what, I focus on personally. So the trying to describe what is RNAi. Again, what are the, messages that might reassure, trying to think about, the regulation of the product that is important. And then trying to, do some testing in terms of what other information might be of interest, to an audience. So this is just a sampling of the questions. There are many others, but hopefully you can get from this overview a bit of the flavor of how in-depth we try to be in this process. So what I'd like to leave you with, and this is really, I think, the primary message is we need to think about the words that we're going to use. And I think Joe was very, very clear in terms of what you have to think about, and staying on what the science tells you. And that's where the sweeping claims, avoiding this technology is going to solve all the problems in agriculture. No, that's not the way you should talk about it. You should think about, it's not the solution. It's part of the toolkit, and it would be, adding to that. And again, trying to give the perception that RNAi is something that's nothing new or well understood. Again, that could potentially, undersells the fact that you're going to have to go through a regulatory process. And so that, again, that's probably something to avoid. Then the focus on the product development from an industry point of view, that's something that we would certainly avoid as a developer. Trying to find the right, product to move into development is difficult, and trying to say that you've got a shortcut, again, way of when you're using RNA-based products, it's probably not the right message. But the final thing that I'd like to focus on is that some of the words that we've used, in literature, actually hurt us, in my view, in the communication. And so words like silencing, no one in a public dialogue likes to be silenced. Being ignored is particularly in today's social media world, is something tantamount to, you're always going to be on the blacklist side. Knocked out, or things like are in the same boat. And when I try to talk about this to general public, which includes my friends and family, I use the dimmer switch, which that's what these are. The photograph at the top is a volume control for a drum kit speaker that we happen to have in our house. And that's more of an simple way of communicating that, yeah, that's really what RNAi is. You're just adjusting the volume of the speaker, or you're using the dimmer switch to turn down the light in the room. and so some of those kinds of images. So that's really the message I'd like to leave with you, is that there are some words that we should think about using, and some words that we should lose. And so thank you for the opportunity again to be with you too today. Hugh, you're muted.
Thanks, Greg. Yeah, I've done this so often, and you would have thought I'd have learned by now how to work this this kit. Yeah, so thank you for your presentation, the importance of vocabulary and the specific words that we use. So there's time remaining for a discussion on this session, and then there's also time in the meeting scheduled for an overall discussion, sort of tying together all of the threads that we've heard from the various sessions, the various speakers in the various sessions. So there's nothing new on the chat, no questions specifically for the speakers in this session. I'll give it another second, but has anybody got anything, anybody online, Bruno, Hildegard, others,
if
they
I
might
may,
want to ask?
if I may, just one thing. Do you, I don't know, Bruno, can we get a collection of all the comments afterwards so that we can read through all of them? Because I haven't had time to do that. Your microphone, Bruno.
Everything is recorded, and so it will be available on the web page. But
okay.
I think that we can still dedicate some time to the discussion.
Yeah.
If there are some questions or some other people that are now connected that want to make questions directly, can just open the video and we can manage the discussion.
So if Bruno could break in again, I think the last presentation that I've had an opportunity to give to iPlanta Group focused on at least ended with a call out to a paper about how RNA fits in sustainability that researchers involved in this project have been, and how important that is in the public dialogue. Again, I think Mark mentioned that it's the regulators and politicians that we really need to reach, and having foundational publications like that to point to help frame the dialogue from scientists that are actually involved in the research, I think is a fundamental point. We in industry have tried to do a better job in the RNA space by publishing most of the data that we have generated in terms of the hazard and risk assessment profiles. So then that's really, again, hopefully provided a foundation that can be used in that space as well. So just want to call that out and encourage it to continue.
Okay.
One thing I was wondering when listening to the presentations is if we should avoid getting linked to GMO, should we then not use the collected name RNAi, but have subgroups that would, where some of them definitely are not GM, but that makes it more complicated again. And as soon as somebody makes the link between RNAi or any RNAi and GMO, then it's too late, or?
Are you directing that to any particular person, Hildegard? Oh, looks like Greg wants to come back on.
Yeah, so I just to add again, we've actually had conversations with the US EPA in this space, and they've asked for, in the topical area where, again, if you've got something like it was talked about earlier, the DV7, which is part of the trait, it's clearly GM, right? So, but when you're talking about topical application, double-stranded RNA, we did have a conversation about what with them and they were sensitive to, we need to be clear in what we're calling this. And so we were able to have that conversation, because again, what regulators will say in public forums about what it is, quote unquote, again, the vocabulary is very important as well. So we're hopeful that, you know, and kind of where we landed was you just call it what it is. It's a double-stranded RNA, you know, and start from there. So.
Yeah, but eventually, sorry if I give my interpretation, and I ask eventually to the communicator people what they think about. First of all, as a scientist, I've been struggling also with some colleagues about this. I fully agree that RNA, like gene editing, it's a GMO. So we are modifying DNA. So we cannot change this. It's what we are doing. Probably we can communicate a little bit different. So for example, now I was revising also some papers, or writing some papers, and on RNA. And instead to keep saying GMO, transgenic, and so on, I say, why not just use stable expression of RNA and external application of RNA? So two different, so use this terminology for differentiate the two technology without entering all the concepts that this is GMO and this is not GMO, because topical application is not GMO. It's almost clear, I think, also to the regulators. So I don't know how far this approach can help in keep a little bit different. So clearly, finally, it's a GMO, but at least we speak a different language. We use a different terminology for defining what is the product and what we are doing. Stable expression and external application. Is that a terminology that can help?
Yeah, so yeah, this is Bill Moore speaking. I wonder if we can draw some examples or learnings from BT. So, I mean, BT is actually used in organic production. You know, you're using this, it's the same proteins that actually kill insects organically with BT proteins versus what's expressed even in a plant transgenically or with the GMO. So can we use any corollaries from that when it comes to RNAi, whether it's expressed in plants or sprayed as a pesticide? So we do the same thing with BTs already.
Okay.
Yeah, that's a neat analogy, in fact. Yeah.
I think Sue posted a question now. I think Marlena's brought up an important point that should be addressed, and that is that these children are being taught at schools that GMO are bad. Yeah, that is an important issue. How to change this? The next books put forward pros and cons, but the cons are what's remembered. So that is the reality. We should be more active against, not against, but at least in showing the situation is different.
So it's this, again, this is Bill again. We've gone to some teaching conferences, and there's actually textbooks that only talk about GMOs in the negative sense. They don't talk about the pros at all. And so, you know, we also need to reach the publishers who actually put this stuff in the books, for which the teachers can actually glean from. So if the teacher is not educated in this at all and is only reading from the books, our sense is they're only going to be getting a negative perception of GMOs. So there's a lot of layers, as Joe was saying before, there's a lot of different layers of communication, and this is yet just another one that teachers are going to rely on. Just like what Hildegard was talking about with that YouTube video, we have presented that a number of times. I think it is absolutely fantastic. And so I think more of those opportunities have to come out.
So just talking about children, I perhaps naively had the assumption that younger people were more kind of tech savvy in general and might be more accepting of innovation in agriculture and biotechnology. And as time went on, as the older kind of generation, that I agree with Mark right from his first talk, you know, those ideas are pretty firmly fixed in people's minds and it's hard to change. But in perhaps 10 or 20 years, as those guys and gals get older and less influential in society, maybe a new generation will be there with different ideas. But it seems that it could be the opposite if children are already forming negative ideas of biotechnology in school.
So Greg, again, I think there was actually along these lines a very interesting survey that's being put together by a group that's US based called Research America. And they've actually looked at subpopulations in terms of in the mist and been asking some of the same questions over a period of time so they could track the flow of understanding and where they place science and importance of science. And actually, startling to me, your ideological orientation as it goes toward politics has a huge impact on how you view science and science communication. And so I think and it also shows you the power of those of us that have lived through the evolution of the web. I thought it would bring people hopefully closer together, but it's what has happened is it divided us into more camps. And we only go to places where, we reinforce our ideas. So I think that's my concern about, if you're a younger person that's never lived for a time when you didn't have one of these in your hand, the way you seek information, the way you process it is very different. So I think that's, I think that's where our challenge lies in that. So thanks.
There's a comment from Jeremy. Do you want to say something, Jeremy, about biology teacher? Well,
yeah, I think this issue of teachers is an interesting one. Unfortunately, in this country and maybe in others, we have a lot of teachers teaching science and biology who are not really biologists or scientists themselves. Often they have qualifications in geography or other areas, economics or whatever. So they therefore tend to present more a sort of societal perspective of GMOs, which often is anti-GMO. On the other hand, I remember working with one brilliant teacher in a school nearby who actually got his students, these were people about 16-year-old, 15, 16-year-old, he got them to collect articles from newspapers and then to do critiques on them. And he got them to look at an article in the newspaper and then to say what was wrong with it, where the science was bad, where the science was good, and so on. And a lot of them actually were GMO-related topics produced by popular newspapers. And it was very interesting to see these kids being analytical, looking very objectively at what journalists were writing and commenting. And I could see what this teacher was trying to do. He was trying to make people aware of the fact that, you should approach things from a scientific perspective and not just believe all the information that's flying around. You should do your own analysis of the information. And I mean, my daughter went to a school where they actually did transformations. At the sixth form, they were doing basic transformations of bacteria and one or two other things, and they were being shown how to do genetic modification. And I thought, that's great. This is what they should be doing. So the perspective of those students was very, very different from students who are not exposed to, if you like, real science and real scientists.
Well, I can add that in addition to that, generally regional government, at least in Italy, and local governments support much more kind of communication program for supporting organic product, organic agriculture, including the fact that sometimes they're forced to use organic product in the mensa, for feeding the children at school. And that is clearly a communication message, important communication message. The children practically, they can eat only organic food because the schools are forced by the local government to buy organic food. And that is, and sometimes some people realize that they have problems because they cannot find enough organic food in the different period that can allow the school to have a good nutritional balance in a good balance in the food that they give to the children. But that is also a strong message given to the children.
Yeah. Well, it concerns me too that some of the organic produce has huge food miles on it. You know, when you see the range of organic, you know, exotic fruits and vegetables and so on, labels as organic in our supermarkets, you just think, is this really organic? You know, it's being air freighted, it's being shipped from tropical countries. You know, it's maybe a commodity crop, and it's depriving the local people of the basic foods that they need to grow themselves. And so I do question sometimes, you know, the definition of what is organic and what they consider to be sustainable organic.
Yes, I agree. But the message is you get organic, then nobody's going to control it.
Yeah.
Well, I don't know if there is other question because the people is leaving. I have a question, maybe a real final question before to thank Mark, Joe, and clearly everybody. But in particular for Mark and Joe as external, how do you feel about this network? Do you think that this kind of network can be useful? So organizing this type of activity, get together scientists, try to discuss about terminology, strategy, and so on. Do you think that may help for doing this, for not saying solving, but changing a little bit the situation, the perceptions that the people has? And finally, do you think that the strategies that we are proposing are appropriate, or should we? You already give us many information, but in general, what is your feeling?
What strategy are you proposing?
No strategy. Just to get together and try to discuss. That is what we need to come out.
Well, I mean, that's an important first step, and I think that if the scientific community and the product developers have been able to do that when they were first developing the GMO traits, maybe we'd be in a different place today. So yes, thinking about it, and as Joe said, learning from the science communication, I think is critically important to get out in advance of the time when this would become potentially a huge public controversy. I think something that came up in the chat earlier is worth taking away from this, because I think it's really, really important. On its own, RNAi does not have the characteristics to become a breakthrough public controversy. I fully agree with that. But if it's associated with GMO and gets lumped into the same basket, then it's game over. It's not going to be possible to rescue the GMO issue. I've tried that, and I know many people on this call have tried that for going on 10 years. If, on the other hand, you know, we've seen several really interesting presentations about how RNAi is potentially a huge breakthrough medical tool for human health. You know, if you can take this out into the public and RNAi becomes seen as, well, you've already got RNA there for COVID vaccines. If it's also an antiviral therapeutic, RNAi gets seen as that. Maybe it's something which can even cure cancer. I mean, then you're in a different place. and the branding, just the name, just what RNA will evoke in people's minds is going to be much more positive if you ever want to use it for agriculture. So let's not be too gloomy about this. I think it's very clear what we should not do. I'd be very I mean, some people have said, well, maybe stable expression is okay, maybe we can get away with that. It's not going to work because it's taken under the regulations as transgenic, and therefore will fall under GMO regulations in Europe and elsewhere. So don't lead with a GMO RNAi products straight away. Do more work on the health and maybe on the biopesticides. Learn from the Impossible Burger. Have more conversations like this, and just make sure that the first product lines which you want to introduce to the public have clear benefits both to public health, to the consumers, and hopefully also to broader society and to the environment.
Great, thank you. That's really useful. Well, yeah, let me, if no other question or comments, I think that we can really stop, and I hope that you, Mark, and other people will keep following us. Maybe we will have other opportunity in the future to do something together, and I hope that it will be possible to maintain this contact and this type of exchange and collaboration. So again, if nothing else is coming up, I want to thank everybody, and particularly Hilde that took over the organization of this event, of this webinar, Guy, Ismaji, and Lou Jones and Jeremy that help us, and clearly all the speakers. So thanks again for joining us today. And if before to close everything, if you open up your camera, I will try to get a picture, a virtual picture. So please.
Like the face band, Greg.
I don't know. Possibly we can try a boy to a boy. So let me just give me a minute. Okay, let me save and have another one. Just to be sure. Okay, great. So thanks, thanks for this. And no, sorry, in the second one, William was off. So, well, people is already going. I don't know if you can turn on the camera. William, are you there, William? Because I see William. I think that he's already left. Okay. By the way, we have this. We have this. Okay. Thank you very much. Have a nice day. Let's stay in touch, and I hope to see you again next in another event. Have a nice day to everyone. So goodbye to everybody.
Thank you, everybody. Bye-bye.
Bye-bye.